All right, welcome back. In today's concept, we're going to talk about an action potential here. And so, what we have is a graph of the action potential. And we'll talk about each of these phases of this graph here. But I guess we'll just start right here at the beginning, what we call a resting membrane, or just what is a membrane potential to start. Remove my toolbar. And the first concept is the idea of a membrane potential. And if you haven't watched the video, I encourage you to watch the video on membrane potentials and what causes membrane potentials. We'll talk about that briefly here. But basically, what you have is that distribution of charges across the cell membrane. And this is actually measured in millivolts. The standard is that we measure the inside of the cell in reference to the outside. So the outside of the cell is always going to be our reference number, or 0, right? It's going to have a reference of 0. And then the inside of the cell is going to have a charge somewhere in the range of -60 to -90 millivolts, right? And so the concentration of ions in the extracellular fluid is largely balanced with a net neutral charge. However, there's a slight difference of charge across the membrane surface, both internally and externally here. And the difference is limited in this region so that all of the information that's generated across the cell membrane really just travels across the cell membrane itself. Now, before we get too far ahead of ourselves, we should describe the state of a resting membrane potential. So at rest, pretty much you have a high concentration of sodium outside of the cell. So circled here in the extracellular space is circled is the extracellular space. You've got a high concentration of sodium outside of the cell here. Particularly you can see resting right there around the cell membrane. On the inside of the cell, you have a high concentration of potassium. Maybe I'll make that a different color here for us. That pink color looks like a good one. You've got a high concentration of potassium here, as well as a high concentration of these large negatively charged proteins, aminos, running around the surface there. So what you get is, again, a charge somewhere in the range of -60 to -90. I'm going to use the number -70 millivolts on the inside relative to the outside of the cell. And the biggest mechanism in maintaining the sodium potassium pump concentrations is, or the sodium potassium concentrations is, the sodium potassium pump. And now recall that the sodium potassium pump is responsible for releasing 3 sodium ions into the extracellular space, the interstitial fluid as it might be called, and 2 potassium ions. So you can see there's a net positive of 1 ion into the extracellular space for every action that occurs, as well as the negatively charged, and I'll put an A here, those negatively charged amino acids that are floating through. And so hopefully we have this idea of a resting membrane potential, right? The excitability or irritable cells of our body, like muscles and nerve cells, have this resting membrane potential, or a charge difference relative to the outside and inside of the cell. And it's good. It's going to be big to know that the concentration of sodium is high outside of the cell, and the concentration of potassium is high inside of the cell. So kind of in relative terms. So at resting membrane potential, we can orient ourselves into this graph, but it's the same thing that we've been looking at. We've got a high concentration of sodium in on the intracellular space, or the interstitial fluid, and the extracellular fluid here. And then the sodium potassium pump, or the sodium potassium transporter here, releasing 3 sodium ions into the extracellular space, while bringing 2 potassium ions into the intracellular space there, the cytoplasm. And so we also have some other channels that we should be familiar with, one of which is a leakage channel, which we can see here. Leakage channels just simply allow ions to float in and out and help to maintain that resting membrane potential. What you also have are what we talked about are voltage gated channels. Now, a voltage gated channel will respond to a change in a membrane potential. We'll talk about this one here in a sec, but let's stick on this idea of resting membrane potential. When the resting membrane potential, this is when a cell is at rest. But if we stimulate a cell, we can stimulate a cell one of a few ways, right? If it's a muscle cell particularly, muscle cells are stimulated by a neurotransmitter. So a neuron will release this neurotransmitter. In the case of muscles, the neurotransmitter is acetylcholine. Acetylcholine binds to the receptor on the protein, and as a result, the channel opens. You can see here ions move across in response to their gradient. So you'll have sodium ions now rushing in. You'll have potassium ions now rushing out. So in response to a muscle cell, you'll have a neuron which will release a neurotransmitter like acetylcholine, binds to the receptors, and now ions can float in and out. Similarly, there are mechanically gated channels, and so the signal that's sent to the brain, the neurons send a signal to the brain for sensory. And so when mechanical changes happen, when I press on my finger here, that changes and deforms the cell membrane of the sensory neurons here, and so that signal will then open channels, allowing for the free flow of ions as well. And again, they're going to move towards their concentration gradient. And so what happens is you get what's called depolarization. Depolarization is the scenario in which, as we've mentioned, the sodium ions are going to move towards their concentration gradient, which is inside of the cell. Potassium ions are going to move towards their concentration gradient, which is outside of the cell. And what you have is a total resting membrane, or a membrane potential, which moves towards 0. And so that's what we have here. At our resting state, you get the initial response. What is that initial response? Well, if it's a sensory neuron, perhaps it's the pressure or touch, like we mentioned. Or if it's a muscle, perhaps it is the stimulus from a neuron. The neurotransmitter has now bound to the muscle, and now we're getting that stimulus. And that's what we see here. The stimulus here, which is going to drive the inside of the charge on the inside of the cell closer to 0. There's 0 right here. Now you can see that this idea is that at negative 55, we hit what's known as a threshold. Now, threshold stimulates a different type of receptor. Threshold will stimulate voltage-gated channels. So you can see here that at rest, we've got a voltage-gated channel at that good old negative 70 millivolts there. But once we hit our threshold, maybe let's say negative 55, negative 50 millivolts, the voltage gated channels open. So where the ligand gated channels demonstrated a small little response, the voltage gated channels, which are numerous across the cell, will then cause a large spike in depolarization. All these gates channels open across the cell membrane and move in such a manner we'll erase these, move in such a manner that it flips very quickly, right? So the gates open, the voltage gated channels open. As a result, sodium rushes into the cell, potassium rushes out of the cell, and you get this flip, and this charge runs right across the cell membrane, and as fast as it opens, it closes. So as fast as it opens, it closes back up. So that at the peak here, we get a charge that's actually positive. The entire thing just flips, right? We went from a negative on the inside to positive on the inside here. And so once it reaches that positive voltage, then we start to close those channels down. Oh, let's go here, where we go back to repolarization. And so in most cells, muscle and nerve cells, what we have is a hyperpolarization. So oftentimes, due to the leakage channels, so leakage channels here are channels which are, where's it? I don't have leakage channels here. Leakage channels are ones that just maintain and stay open. They, as a response, they maintain open just a little bit longer, particularly potassium channels as well are a little bit delayed in closing, and as a result, you get a little bit of an overshoot. And this hyperpolarization allows for a little bit of a break in those cells, right? So what we get is now there's resting membrane potential. It goes a little bit below resting membrane potential. It requires more of a stimulus to get to this action potential. And so the idea of an action potential is that it's an all or nothing principle, meaning that if you do not reach a threshold, you do not get an action potential. It's simply a graded potential. And so once you get that stimulus to the action potential, you have a respondent depolarization and oftentimes a hyperpolarization, a overshooting, which allows for a break in the muscle. And so here's the chart that we just went over here, right? We see that there is the resting membrane potential. This could be a neuron. This could be a muscle in the case here. So then we have the stimulus that's applied. The ligand gated channels start to open here. The stimulus could be either sensory pressure or it could be the neuron stimulating a muscle here. And that stimulus causes, and again, this is causes a little bit of a change. If that change is great enough, we will stimulate voltage gated channels at this point here. And once voltage gated channels open, we get a large depolarization to positive, and then quickly repolarization, which then would result in hyperpolarization, which allows muscles and nerves to take a little bit of a break in between the next contraction and or stimulus. And so this is that chart, except this time we're looking at the reference of these two. We've got a motor neuron, so this is a neuron here. You've got a skeletal muscle, that's a muscle, and a cardiac muscle, right? So you're looking at three different types of muscles and the action potentials that are provided as a result. So this is time on the x-axis. On the y-axis, you've got membrane potential, transmembrane potential there. And so if we were to, let me clear this up here just a little bit. If we're to see here, you see there's your resting membrane potential. There is the kind of the point of that slow depolarization. Then you've got rapid depolarization and then repolarization here down towards a little bit of a towards our hyperpolarization, right? So if this is our resting membrane, you've got hyperpolarization that sits right there. Now the skeletal muscles have a resting membrane potential at about negative 90 millivolts here. Then again, there's the kind of that slow depolarization, rapid depolarization, and then repolarization. And then the same thing happens with our cardiac muscle here, but you can see that repolarization takes a little bit longer kind of as it starts. And again, it's because one of those things is that cardiac muscle is involuntary, right? So we don't get that quick stimulus and break, and it is the function of the cardiac muscle to be a little bit more slower and deliberate, right? But that's the concept of an action potential in just under 20 minutes. Hopefully that was helpful. We'll continue to talk about the action potential as it relates to a skeletal muscle as well as the nervous system, the neuron, in further sections. But just wanted to introduce the concept of membrane potential and action potential here today. I wish you all guys the best. Thanks for your time.